Polymerase chain reaction, PCR, principle, procedure or steps, types and application. Principle. Polymerase chain reaction is method for amplifying particular segments of DNA. It is an enzymatic method and carried out in vitro. PCR technique was developed by Kary Mullis in 1983. PCR is very simple, inexpensive technique for characterization, analysis and synthesis of specific fragments of DNA or RNA from virtually any living organisms. Steps or procedures. PCR consists of 3 basic steps. 1. Denaturation. 2 strands of DNA separates, melt down, to form single-stranded DNA. This step is generally carried out at 92°C-96°C for 2 minutes. 2. Annealing. Annealing of primer to each strand is carried out at 45°C-55°C. Extension. DNA polymerase adds DNTPs complementary to template strands at 3 feet end of primer. It is carried out at temperature of 72°C. Factors affecting PCR. I. Primer. PCR reaction needs 2 primer, a forward and a reverse primer. Primer are synthesized oligonucleotide usually ranging from 15 to 30 bases long. Primers are designed such that at 3 feet end they do not have more than 2 bases complementary to each other, as this results in primer-dimer formation. The G+C contents is in the range of 40 to 60%. The melting temperature, Tm, of both forward and reverse primer is usually the same. Low concentration of primer results in poor yield, while high concentration may result in non-specific amplification. Hence, optimal concentration of primer is needed, i.e. 0.1 to 1 micro. E. Amount of template DNA. Optimal amount of template DNA usually in nanogram. Higher concentration inhibit or results in non-specific amplification. toc-DNA polymerase. toc-DNA polymerase is 94 kD thermostable DNA polymerase isolated from Thermus aquaticus. Optimal temperature for activity of toc polymerase is 72 degrees, but it can tolerate high temperature and do not effects by denaturating temperature of 94 degrees Celsius. toc-DNA polymerase have both 5'-3 polymerase activity and 5'-3 exonuclease activity. But it lacks 3'-5 exonuclease activity, proofreading activity. Types of PCR. 1. Standard PCR. Nested PCR. Random amplified polymorphic DNA. Long PCR. Restriction fragment length polymorphism, RFLP. Amplified fragment length polymorphism, AFLP. Multiplex PCR. Single cell PCR. Fast cycling PCR. In situ PCR. High fidelity PCR. Asymmetric PCR. Repetitive sequence-based PCR. Overlap extension PCR. Assemble PCR. Mini primer PCR. Solid phase PCR. Touchdown PCR. 2. Reverse transcriptase polymerase chain reaction, RT-PCR, for RNA. 1-step RT-PCR. 2-step RT-PCR. 3. Real-time PCR, for DNA or RNA. Dye binding to dsDNA. Fluorescent probes. Application. Forensic science. DNA fingerprinting, paternity testing and criminal identification. Diagnosis. Molecular identification of microorganisms. Evolution study, evolutionary biology. Fossil study, paleontology. Gene cloning and expression. Gene sequencing. Vaccine production by recombinant DNA technology. Drug discovery.